दिस वीडियो वी विल डिस्कस अबाउट द डीएनए स्ट्रक्चर इन फंक्शन। इस वीडियो में हम डीएनए स्ट्रक्चर मतलब इसकी साख और फंक्शन मतलब इसके अफ़ाल के बारे में पढ़ेंगे। सबसे पहले हम डीएनए के बारे में इंटरेस्टिंग इंफॉर्मेशन सबसे पहले मैं आपके साथ शेयर करता हूँ, फिर हम अपने टॉपिक की तरफ जाते हैं डीएनए स्ट्रक्चर की और इसके फिर फंक्शन के बारे में। डीएनए इज द डेटा बैंक ऑफ लिविंग ऑर्गेनिज्म। डीएनए मॉलिक्यूल कंटेन द मास्टर प्लान ऑफ ऑल कैरेक्टर ऑफ एवरी लिविंग ऑर्गेनिज्म। मतलब इंसानी जिस्म में जितनी भी खुसूसियात है इन सारे को कंट्रोल करने वाला हमारे साथ डीएनए है। ह्यूमन डीएनए होल्ड ऑल द इंफॉर्मेशन अबाउट द ह्यूमन बॉडी, ऑल द डेटा सच इज द कलर ऑफ हेयर। मिसाल के तौर पर हमारे साथ हेयर का कलर किस तरह होगा, स्किन और आई इसकी कलर किस तरह होगी, द हाइट ऑफ द बॉडी, द साइज, अपीयरेंस, मॉर्फोलॉजी, नोज, फिंगर, ईयर, eyes, etc. We can say each and every character is controlled by the DNA. There is a lot of information stored in the DNA. DNA में बहुत ज़्यादा मालूमात स्टोर करने की सलाहियत होती है। If we had to write down all the information in the DNA, अगर हम DNA पर मौजूद मालूमात को अगर हम राइट कर ले लिखें, it would make 900 वॉल्यूम। तो इससे हमारे साथ तकरीबन 900 वॉल्यूम की बुक्स राइटिंग में आ जाएगी, each of 500 pages. So we can say that a total of 4,50,000 pages the information is written from a single DNA molecule. So all this knowledge has been fitted into a teeny molecule that we cannot see with our naked eyes. ये जितनी भी मालूमात, जिस तरह मैंने कहा कि 4,50,000 पेजेस पर जितनी मालूमात आती है, इतनी मालूमात ऐसे एक छोटे से मॉलिक्यूल में मौजूद होता है जिसको हम अपनी आंखों के जरिए नहीं देख सकते। Who has fitted all this information in DNA? Certainly this is the work of supreme intelligence. He is Allah with the supreme intelligence, limitless knowledge and eternal power, who created the universe. Now we'll go to explain what is the DNA. DNA, a polymer of deoxyribonucleic, is known as a DNA. DNA is found in chromosome, mitochondria and chloroplast. It carries the genetic information from generation to generation. Or we can say that nucleic acids are polynucleotide, or we can say that DNA is a polymer formed by the linkage of units called monomer known as nucleotide. And we can say that there are two types of nucleic acid, that is DNA, which is known as deoxyribonucleic acid, and RNA, which is known as ribonucleic acids. Each nucleotide is basically composed of three components: a nitrogenous base, a 5-carbon sugar known as pentose sugar, and a phosphate group. So this is basically the component of a nucleotide. And we can say in a typical nucleotide, the nitrogenous base is attached to carbon number 1 of pentose sugar, while phosphate group is attached to carbon number 5 of the pentose sugar. The bond formed between the phosphoric acid and hydroxyl group of pentose sugar is called ester linkage. What is the difference between nucleoside and nucleotide? When a nitrogenous base, adenine, thymine, guanine, cytosine or uracil, when one of them is attached to a pentose sugar, nucleosides are produced. And when this nucleoside attached with a phosphoric acid or a phosphate group, nucleotides are produced. So in nucleoside 2 component is present, while in nucleotide 3 components are present. So basic structure of pyrimidine and purine. Pyrimidine is a single ring nitrogenous structure and purine is a double ring structure. This is the main difference between pyrimidine and purine. The examples are pyrimidines are cytosine, thymine and uracil. Cytosine is common in both DNA and RNA. Thymine is present only in DNA and uracil is only present in RNA. Purine is a double ring nitrogenous basis is known as purine, and their example is adenine and guanine, and both of them they are present in DNA as well as in RNA. The various types of nucleosides and nucleotide which are present in DNA and RNA, they are present in the form of purine and pyrimidine, single ring structure as well as in double ring structures. Nucleosides and base analog can be used as anti-cancer and antivirus drugs. For example, those chemical which is used against cancer, they are known as anti-cancer agents. Their example is a 6-mercaptopurine and 5-fluorouracil. These are various types of chemical which is used as a anti-cancer. Antiretroviral agent, antimine against retroamine RNA containing a virus which cause various types of diseases. So their example is azidothioamidine and dideoxyribonucleic. These are the example of antiretroviral agents. Now we will come to explain what is DNA structures. The primary structure of DNA is in the sequence. Here you can say that at 5 end, phosphate group here is a pre, while at 3 end, hydroxyl group is a pre. Here we can say that a pentose sugar, their phosphate group is pre, while here in pentose sugar, hydroxyl group is a pre. And the bond which is produced between one nucleotide and another nucleotide is known as phosphodiester linkage. This is the primary structure of the DNA. Traditionally, a DNA sequence is drawn from 5 to 3 end. For example, if we take the example of this type of DNA, so a shorthand notation for this sequence is adenine, cytosine, guanine, thymine, and adenine. And the secondary structure of DNA is the double helix. You can see clearly in the diagram. The secondary structure of DNA, two anti-parallel polynucleotide chain would around the same axis. Sugar phosphate chain wrap around the periphery, and bases adenine, thymine, cytosine, and guanine occupy the core, forming complementary. That according to the Watson and Crick model of DNA structure, that adenine will always attach to thymine and guanine will always attach to the cytosine, vice versa. The DNA double helix is held together mainly by hydrogen bond. So a hydrogen bond, a chemical bond in which a hydrogen atom of one molecule is attracted to an electronegative atom, especially a nitrogen, oxygen or chlorine atom, usually of another molecule, is known as a hydrogen bond. So you can clearly see that between the adenine and thymine nitrogenous bases, two hydrogen bond is present, while in guanine and cytosine, three hydrogen bonds are produced. So two hydrogen bond between adenine and thymine pairs and three hydrogen bond between cytosine and guanine paired. You clearly see here in the structure of adenine, thymine and cytosine and guanine. Base taking. The bases in DNA. The bases in DNA are planar and have a tendency to stake. Major staking forces are hydrophobic interaction and van der Waals forces. So we can say here that the bases are projected to one side of the strand. The sequence of the nucleotide in one type of DNA is constant while it is different from other type of DNA molecule. So DNA molecule consists of two strands and the two strands twist about one another in the form of a double helix. The two strands run in opposite direction to each other in the double helix. They are held together by hydrogen bond. So structural form of DNA. Based on the structure, there are various types of DNA: A DNA, B DNA, and Z DNA. And their properties, for example, handedness, base pair per turn, rise per base, etc., are given in this table. Normally, a hydrated DNA is the B form of DNA. We can say that normal DNA, their structure is a B form. And their helical sense is right-handed. And their base pair is present almost. And they are perpendicular to the helix axis. And they produce 3.4 angstrom apart. That both the strands are separated at a distance of 3.4 angstrom. One turn of the helix is 36 angstrom or 10.4 base pairs. Minor groove is 12 angstrom and major groove is 22 angstrom. You can say that two grooves are present within the DNA molecule, minor groove and major groove. In eukaryotic cell, DNA is folded into chromatin. DNA tertiary structures. The DNA double helical structure coil around histone. DNA bound to histone form nucleosomes, for example, and their thickness is 10 nanometers fibers. Nucleosome contain 146 nucleotide. So nucleosome, what is nucleosome? Any of the repeating globular subunit of chromatin that consists of a complex of DNA and histone, and it is composed of 146 nitrogenous base nucleotides. So histone cover of nucleosome, and these are the linker DNA. So it just like beads on string. So structure of a nucleosome cover. Four pairs of histone proteins are present: H2A, H2B, H4, and H3, and they form 8 protein within the histone, or we can say nucleosome cover. So here DNA, when it is present, beads on a string form of chromatin and 30 nanometer fiber, then one loop is equal to 75,000 base pair, one rosette is equal to 6 loops, and one coil is basically composed of 30 rosettes, and two chromatids is basically composed of 10 coil each other. Supercoil, coil over coil. You know that when the simple form of DNA, when they react with various types of protein, and protein plus DNA, they produce the chromosome. And chromosome, we can say it is a very complex structure. It is produced due to the coiling, again coiling, ultra coiling, super coiling, and it produces a structure known as chromosomes. What is DNA melting and annealing? When we heat a double-stranded DNA molecule, it is converted into single-stranded DNA molecule, and this process is known as denaturation. And when, for example, the condition is suitable, then again the nitrogenous bases, they form hydrogen bond, and they again converted into double-stranded DNA, which is known as annealing. Melting point of DNA, the temperature at the midpoint of the transition, you can see clearly here in the table. The content of the guanine cytosine base pair, it depends upon the size of DNA, pH, as well as ionic strength. Function of DNA and summary of the structure. DNA consists of four bases: adenine, guanine, cytosine, and thymine, that are held in linear array by phosphodiester bond through the 3' and 5' position of adjacent deoxyribose moieties. DNA is organized into two strands by the pairing of bases adenine to thymine and guanine to cytosine on complementary strand. These strands form a double helix around the central axis. The 3 into 10 power 9 base pairs of DNA in humans are organized into a haploid complement of 23 chromosomes. And DNA contain, or we can say DNA provides a template for its own replication, and thus maintenance of the genotype, and for the transcription of the roughly 30,000 human genes into a variety of RNA molecules. So the synthesis of DNA from DNA is known as DNA replication, and the synthesis of various types of RNA from DNA is known as transcription, and the synthesis of protein from messenger RNA is known as translation. And this whole process is known as central dogma of molecular genetics. If you like the video, please share, subscribe, and